Hello everyone, we are group 21. We are going to explain MHC restriction. In simple words, MHC restriction means that a T cell can interact with the MHC and the peptide bound to it, but only recognize the antigen when it is bound to the correct type of MHC molecule. What is MHC? MHC molecules are the molecule that appears on a cell surface and is used for identification of self or non-self. The genes encoding this are unique individually, like a business card, so each human being has his own MHC. MHC is divided into class 1 and class 2. All nucleated cells have MHC class 1, while only antigen-presenting cells, such as dendritic cells and macrophages, have class 2. Each T cell has a unique antigen-recognizing receptor called TCR on its surface. T cells use TCR to recognize a peptide antigen. However, it can't recognize an antigen itself, but can recognize an antigen presented on MHC molecule.
There are two major subtypes in T cells. One is CD8 positive T cell. CD8 positive T cells have membrane glycoprotein called CD8. That's why they are named CD8 positive T cell. Each CD8 positive T cell have a TCR, which can recognize only one type of antigen.
The CD8 positive cell works as a cytotoxic T cell, which means it kills infected or damaged or tumor cells. When cells are infected or damaged, they present antigen on their MHC1, not MHC2, and it kills them after it identifies self-MHC1 and connects the MHC1 antigen complex, which its TCR fits.
Let's go into more details. After a mouse is infected with a virus, cytotoxic T lymphocytes, the activated CD8 positive cells against the virus, can be seen a lot in the animal. The target cell from the same mouse, strain A, infected with the virus, presents MHC1 of strain A and the peptide antigen of the virus on its surface. After mixing this target cell from strain A and the virus-specific CTL from strain A, the CTL can recognize self-MHC1 and the peptide and kill the target cell. The target cell from another mouse, strain B, which means no blood relationship with strain A, presents MHC1 of strain B and the peptide antigen of the virus. After mixing this target cell from strain B and the virus-specific CTL from strain A, the CTL cannot recognize the peptide because it is presented by allergenic MHC1 of strain B.
A CD4 positive cell works as a T helper cell. It recognizes the enemies and tells the lymphocytes to attack them. An antigen-presenting cell, APC, processes the pathogen into peptide fragment in some cases, such as when it takes in bacterium, and presents a peptide antigen to MHC class 2, not MHC1. The TCR of a CD4 positive T cell only recognizes the peptide antigen when it is a self-MHC2 molecule plus antigen. When a CD4 positive cell receives the information and gets activated, it divides and instructs lymphocytes to eliminate the enemies.
Like this, which T cells can connect to autologous MHC1 or MHC2, presenting viral antigen is determined. This system is called MHC restriction.